All right, in this explainer, we're going to dive deep into a really interesting new paper from Shi and colleagues. It gives us a powerful new way to look at the side effects of EGFR tyrosine kinase inhibitors. We're going to break down their unique dual methodology approach and see how it really helps us, in the clinic, navigate the pretty complex world of TKI toxicities. So this really gets to the heart of the matter, right? It's the core clinical dilemma we all face. Picture this. It's Monday morning, your patient with EGFR-mutated lung cancer is in front of you, and you've got this whole arsenal of really effective drugs. But efficacy isn't the whole story. How do you actually weigh those nuanced, tricky safety profiles to make the absolute best, most personalized choice? Well, that's exactly the question this paper is going to help us answer. Okay, so here's our roadmap for today. We'll kick things off with that clinical dilemma. Then we'll look at the study's really clever two-lens methodology. After that, we'll break down the key findings and finally, translate it all into what this actually means for your clinic. Let's get into it. All right, first up, the clinical dilemma. Look, we all know the classic EGFR TKI side effects, right? Diarrhea and rash are practically gospel. But to really do right by our patients, we have to move beyond that. We need a much more granular, a much more comparative view of the risks across this entire class of drugs.
And here's the thing. The simple fact is, we've got tons of data from all the individual big trials, right? But what we don't have are many direct head-to-head safety comparisons across all the available TKIs. And that creates this huge knowledge gap, right where we need the most clarity for making personalized decisions. That's the gap this paper really tries to fill.
Okay, section 2, two lenses. Now, this is what I think makes this paper so compelling. The authors didn't just use one method. They used two totally distinct but complementary approaches to look at drug safety. And what you get is this uniquely robust, really comprehensive picture. So let's break down how these two lenses actually work together. On one side, you have the network meta-analysis, the NMA. This pulls together data from 46 high-quality, randomized controlled trials. Think of this as your super clean, controlled lab coat data. Then on the other side, you have the disproportionality analysis, or DA. This thing mines the massive real-world FERS database. Now this is your messy, boots-on-the-ground data, but its sheer scale is absolutely perfect for sniffing out those rare but critical safety signals. It really is the best of both worlds.
All right, now for the part we've all been waiting for. Section 3, the key findings. You can really think of this next part as the TKI toxicity scorecard. We're about to see exactly how these drugs stack up against each other based on this incredible dual analysis.
So first up, let's take a look at the big picture, the 30,000-foot view from the network meta-analysis. And what it shows is a really clear hierarchy for overall all-grade toxicity. Topping the list, maybe not a huge surprise, is afatinib. It comes out as the most toxic, followed pretty closely by its second-gen cousin dacomitinib. And then way down at the other end of the spectrum, you see the first-gen drugs like gefitinib and icotiniib looking a whole lot better from an overall toxicity standpoint.
But you and I both know that a single overall toxicity score doesn't really tell the whole story, does it? It doesn't give us that clinical nuance we desperately need. The really crucial point here is that each of these TKIs has its own unique adverse event personality. And if we're going to choose the right drug for the right patient, we have to get to know these personalities.
So let's start with afatinib. This is a perfect example where both data sets, the clean and the messy, point to the exact same conclusion. The trial data, it flags the highest risk for oral mucositis. The real-world data, it absolutely confirms this, showing the strongest signals for GI disorders. And get this, a reporting odds ratio for nausea and vomiting of over 21. I mean, that is just a massive signal. It confirms what we all see in the clinic every day. Afatinib's personality is all about that GI and mucosal hit.
Okay, now for osimertinib. And this is really a tale of two different systems. You look at the trial data from the enema, and it flags exactly what you'd probably expect. Hematologic toxicity, right? Highest rates of leukopenia and thrombocytopenia. But then you look at the massive real-world data, and the strongest signal that pops out is for cardiac disorders. And that is such a critical finding, a signal that you really only see when you look at this kind of large-scale real-world evidence.
And speaking of potentially life-threatening signals, the real-world data also screams at us about the risk of interstitial lung disease, or ILD. Now, look, we know this is a class-wide risk, but the real-world data shows an incredibly powerful signal for gefitinib and a really strong signal for osimertinib too. And given how devastating ILD can be, this is a risk we absolutely have to stay vigilant for.
You know, one of the most crucial takeaways for me wasn't just what happens, but when it happens. This quote from the paper really drives home a vital clinical pattern. With afatinib, the side effects hit fast and early, a median onset of just 14 days. But with osimertinib, it's a much more delayed fuse, with a median onset of 70 days. And that has huge implications for how we monitor our patients.
And this chart, it just visualizes that danger perfectly. Look, the GI issues, the ones we see with drugs like afatinib, they show up early. A median onset of 21 days. We're ready for them. Our patients are ready for them. But those cardiac events, the ones strongly linked to osimertinib, they have a much more delayed onset. This is the key point. It means we absolutely cannot let our guard down after that first month of therapy. The risk is still there.
Okay, so what do we do with all of this incredible data? That brings us to section 4, from data to your clinic. Let's really translate these findings into how this could actually change what we do on Monday morning.
What's great is that this research gives us direct answers for our daily clinical questions. Think about patient selection. Afatinib's really intense early GI toxicity, that might just be a deal breaker for a more frail patient. When it comes to monitoring, this data is a powerful call to action for extended cardiac surveillance on osimertinib. It also totally changes how we counsel our patients about what to expect, and just as importantly, when to expect it. And of course, it just sharpens our suspicion for ILD in anyone on these drugs with new respiratory symptoms.
And if you need any more convincing about just how serious that delayed cardiac signal is, here's the number that should do it. In that massive FAERS database, the mortality rate linked to reports of cardiac disorders was over 36%. That's the highest of any system organ class. So this isn't just some statistical signal, it's a life-threatening reality that absolutely demands our proactive attention.
Okay, let's bring this all home. Section 5, the clinical bottom line. If you remember nothing else, here are the 3 most essential, practice-changing takeaways from this whole analysis.
All right, here we go. Takeaway number 1. We need to intensify and prolong cardiac monitoring for our patients on osimertinib. That delayed, high-mortality signal from the real world is just too strong and too dangerous to ignore. Number 2, we have to counsel our patients on the timing of these side effects. Preparing them for early GI issues with afatinib versus the potential for later, more serious issues with osimertinib is key. And finally, number 3, let's really use these nuanced toxicity personalities to personalize TKI selection. It's not just about efficacy, it's about the patient's comorbidities and their quality of life priorities.
And so I'll leave you with this final thought, a question to take back to your own practice. Now that you're armed with this incredibly powerful data on the delayed but very serious cardiac and ILD risks, how will you change the conversation with your very next patient who's about to start an EGFR TKI?